This is Department of Botany, Kalna College. Myself, Dr. Somnath Mondal. Today's topic is Introduction to Plant Biotechnology, and I would like to divide this particular topic into more than one parts. Today we'll discuss the part 1, the basic. Now, this is an overall scheme of transferring a gene from plant A into plant B. Now, the gene of interest present in the DNA of plant A can be either copied by PCR and then eluted, or the gene sequence can be synthesized by oligonucleotide synthesizer. Then, the gene of interest can be incorporated into plasmid vector DNA. This plasmid vector DNA with gene of interest is transformed within E. coli bacteria, and the E. coli bacterium with transformed with our gene of interest and plasmid vector containing the gene of interest can be isolated and again transformed into another bacterium, that is Agrobacterium tumefaciens, which one is the natural genetic engineer of plant. Now, by Agrobacterium tumefaciens, that particular gene of interest can be inserted into plant B. Now, as we know, plant biotechnology and plant tissue culture goes hands in hands, and both are parallel systems and both has to be done parallelly for a successful gene transfer event. In this PowerPoint, we will study some basics of plant biotechnology. We will study two of the very basic tools of recombinant DNA technology, by which a gene can be incorporated into a bacterium. It can be cloned into a bacterium. Now, these two tools are restriction enzymes and plasmid DNA. Now, restriction enzymes were worked upon and discovered by Werner Arber, Hamilton Smith, Daniel Nathans, and they got a Nobel Prize in Physiology in the year 1978 for their research on restriction endonucleases. Restriction endonucleases cut the phosphodiester bond that joins adjacent nucleotides in a DNA molecule, and thus it makes internal cuts into a DNA molecule. Within bacteria, they provide protection against viral infections as they cleave viral nucleic acids and nullify their pathogenicity. Now, restriction enzymes recognize specific palindromic DNA sequences which read same forward and backward on opposite strands of DNA. These are some common palindromic words like madam, reviver, level, civic, which read the same forward or backward. Some similar DNA sequences or palindromic DNA sequences are these: GAA-TTC 5' to 3', and that reads the same on the other strand of DNA, also 5' to 3', GAA-TTC. And this is another example, 5' to 3', GGA-TCC. Again on the other strand, 5' to 3', GGA-TCC. So restriction enzymes specifically recognize these sort of DNA sequences. Now, there are 5 different types of restriction endonuclease. Type 1 cuts at sites away from the recognition site. It is a multifunctional protein which has both endonuclease as well as DNA methylase activity. Type 2 endonuclease cuts within the recognition site or in very short distance from the recognition site. That is why it is very efficient in recombinant DNA technology. and it is an unifunctional enzyme. That is, it has only the endonuclease activity, whereas type 3 cuts at sites in short distance from recognition site. It is also a multifunctional protein with endonuclease activity as well as modified methylase activity. Type 4, they target modified DNA like methylated DNA. Type 5, they use guide RNAs to identify specific non-palindromic sequences and cut the DNA. Now, how they got their names, that is the nomenclature of restriction endonucleases. The first letter of the enzyme denotes the first letter of the genus of bacterium wherefrom it is isolated. Second and third letter of the enzyme are first two letters of species of the bacterium wherefrom it is isolated. The fourth letter is the serotype or strain of the bacterium wherefrom it is isolated. The numbering is done as per the order of discovery of such enzymes from the same host. If it is the first such enzyme discovered, it will be 1. If second, it will be 2. For example, EcoR1, it is isolated from Escherichia coli, and thus the name suggests Eco. Strain RY13R, and it is the first such isolated enzyme from that bacterium. That is why it is 1. Now, there are two types of restriction endonucleases. One are sticky end cutters and another are blunt end cutters. Sticky end cutters like EcoR1, they will cleave the DNA sequence at palindromes and leave some sticky ends or DNA overhanging structures. As we can see in this image, that here EcoR1 cuts in between G and A on either strands and leaves the TTAA, this sequence, overhanging. Now this has the base pairing potential with any incoming single-stranded DNA having complementary sequences. So that is why this is very useful in recombinant DNA technology. And another one is blunt end cutter. For example, SMA1 is such an endonuclease which cuts some blunt ends, as we can see from the image. Now coming to the next tool, that is the plasmid vector DNAs. Now the plasmid DNAs are small extrachromosomal DNAs found within bacteria. They can be approximately 1 to 4 kb in size. They can replicate independent of bacterial chromosome, and they can be used as vectors or carriers to carry our gene of interest into the bacterium by bacterial transformation process. Now, a proper DNA cloning vector has to have following sites, that is origin of replication site or ORI site that will help it to replicate to increase its number, a multiple cloning site or MCS, that is recognition site for several restriction enzymes, selectable marker genes that will allow to select the transformed colonies, RNA polymerase promoter sequences that will be used by RNA polymerase for transcription in vitro or in vivo, and DNA sequencing primers. Now coming to some diagrammatic explanations. Suppose this is a vector map, that is this is a plasmid DNA. Now it has to have an ORI site, that is the site for replication independent of bacterial chromosome. It has to have bacterial selection marker. It has to have plant selection marker. Then RNA polymerase promoter site, left border tDNA and right border tDNA sequences we will discuss in part 2, when we will discuss about Agrobacterium tumefaciens, more of about the transferred DNA, and it has to have multiple sites or multiple restriction sites. Now here each arrow pointing a specific restriction endonuclease will have the cut site, the palindromic site of that specific enzyme. So that is a complete vector map for a proper plasmid vector that can be used to clone our gene of interest. Now the ORI site after bacterial transformation will increase the copy number of our gene of interest inserted into it. Now, how the gene of interest can be inserted into a vector? Suppose the gene of interest can be present within a linear DNA molecule or a circular DNA molecule. If it is flanked with EcoR1 and BAMH1 restriction sites, for example, our plasmid DNA molecule has to be cut with the same set of restriction enzymes to cut it open. Next, the gene of interest with overhanging sticky ends and the plasmid vector cut with the same set of restriction enzymes will be ligated, and the gene of interest will be inserted into the plasmid vector. Now, these plasmid vector carrying our gene of interest will be transformed into bacteria. Now, after bacterial transformation, there can be a number of possibilities. Some bacteria can have our gene of interest along with the plasmid. Some bacteria can have only the plasmid, no gene of interest, and some bacteria can have no plasmid, no gene of interest at all. Now, how they can be separated and separately selected in bacterial culture medium? For that, culture medium are prepared with Luria Bertani agar or LBA medium with kanamycin antibiotic, for example, and another substrate for the bacteria that will on which the bacteria will survive, that is the X-gal, the galactose. Now, if the bacteria has no plasmid, now it will not survive on the bacterial plate having kanamycin antibiotic, because the plasmid has the kanamycin resistant gene. Now, that is the importance of a marker. Now, kanamycin resistant gene is used as a marker that separates between a transformed bacterial cell and untransformed bacterial cell. Now, among transformed bacterial cell, there can be some positive transformation or there can be some false transformation with only the plasmid. Now, there is another gene, that is LAC-Z. Now, this LAC-Z gene will determine whether that plasmid has gene of interest or not. If there is no gene of interest, the bacterial colony will be blue, and if there is gene of interest, it will be white. Now, why is it so? This is because that reason LAC-Z will produce beta-galactosidase enzyme in its active form, and this enzyme will cleave the substrate X-gal into 5-bromo-4-chloroindoxyl, and dimerization and oxidation of this compound will lead to the formation of a blue colored compound. However, if gene of interest is incorporated into the LAC-Z gene and LAC-Z gene will be disturbed, there will be no functional enzyme and thus no blue colored compound or dibromo-dichloroindigo will be developed, and thus the bacterial colony will be white in color. This is called the blue-white screening. Now, from the following bacterial plate, what's your choice? Blue or white? What do you say? Which one contains the gene of interest? So this ends our part 1.